Good evening, everyone. Good evening, everybody. We are waiting for the speaker. It's going to join us very soon. So just a few minutes, and he should be with us. Okay, before the speaker comes in, if you would like to introduce yourself and just share your experience on HackBio with your colleagues, with friends that you've met. Everybody's from HackBio here. Kindly raise your hand. I would unmute you, and you can speak. It's completely optional. If you just want to speak and talk to others. Okay, nobody's raising a hand. Okay, maybe I'll call some people. I actually want to know some people more about the research. The first person that responded to my chat, to my call, was Maruf Ahmed Boulian. I'm going to unmute you so
Yeah.
you can.
So hi everyone.
Yeah, you can speak.
Good evening. Good evening, Wally, and good evening, every participant. So first of all, thank you, Wally, and the total organizing team for organizing such a nice internship program. So I have learned so much from all of my teammates. So since this is a very diverse group of people, this was very interesting to learn different perspective and different opinions, and from different insights from each and every member brings from their own field. So far this has been a very pleasant experience for me, and I have learned so much from each and every team member. And I am looking forward to learning so much more from this internship program. So yeah, thank you.
Yeah, you're welcome. Uh, I want to ask before you mute yourself.
Good.
Like how many countries have you networked with so far?
Uh, so I think I networked with all of my team members pretty well. So maybe
So
6 or 7 countries. Nigeria, one of my team members. Few of my team members are from Nigeria and Kenya, India,
okay.
Pakistan, Bangladesh.
Oh, that's great. Which country are you from?
I'm from
Before
Bangladesh.
we
Yeah.
from Bangladesh. Oh, great. Okay, so we'll not be able to go more than this. Our speaker is around, so I'm going to meet everybody, and then I will unmute Dr. Surajit.
Okay.
Yeah, there is a way we do it. I will first of all introduce you briefly. Okay, Dr. Surajit is our distinguished mentor for proteomics and more in vaccine design and proteomics. So he is the head of the Department of Medical Laboratory Technology and the PI Unat Bharat Habian, Government of India Women's Polytechnic, India. So he's a specialist in vaccine design, bioinformatics. And from our previous chat, I think he has once been a medical doctor. So Dr. Surajit, you have the floor now. You can begin to talk to us.
Okay. Thank you, Ale, for the introduction. Mm, since I am thankful to the HackBio team, which has given this opportunity to interact with a variety of students of different backgrounds and different continents as well. It has been a really nice experience. Although it is just the beginning, we are in the middle of the whole game. As far as I understand, this chat event or this meeting has been designed to interact with the different people and to let them know how exactly this collaborative work can help, I guess. Now it is since the for last 6 months or more, the world is having this trouble of this pandemic. Although these pandemics are not new for human being, this particular the present event has been instrumental uh, to 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 change the way we were doing academics, business, industry, and then what else? Travel, education, upgrading one's skill, and every other thing you can name it. Everything has been a bit changed the way it has been all these days. So I think that this COVID-19 has posed to us a opportunity, at the same time a huge challenge. Now the major challenge for a science worker, for me, I think the major challenge in coming years for those who are doing their studies, for those who are about to work or start their work, the major challenge in coming days in all economies, in developing economies, in developed economies, or in underdeveloped economies as well, the major challenge will be funding for education. What will happen is when all on a sudden something goes wrong, which has a huge impact, then usually it has been seen that the funding for those mm, those domains where the return is not instant usually suffers from a lack of funding. So I expect and I anticipate that there will be major cut in the budget for education, for training, for research, for fundamental research, and those kind of research which generally do not expect to give instant results. In those cases, funding will be very limited. At the same time, the opportunities are also there. The opportunity is people will now um, look at a biologist with more respect, because they'll come to know that it is only the people who dealt with biology. It could be a medical doctor, it could be a teacher, it could be a bioinformatician as well. If somebody have to do something related to this disease, it would be a biologist. So therefore, there will be a renewed interest in biology, that is for sure, and I anticipate that. For last 12 years, I am studying. I am letting people to study. I am training people. I am training myself in various aspects of biological science. Uh, Although the major part of my training was been wet lab experiments, wet lab trainings, clinical, and then where we handle a lot of biological samples. And I should also like to tell you, the region I belong to is the northeastern part of India. It's a semi-urban, or if not rural, it's a semi-urban area, where in a small part, in a small state, there are multiple medical colleges and multiple healthcare facilities. And I must have the, since I'm having the opportunity, I'm just sharing with you that as I think I have already told this to the other interns as well, that approximately 150 students of mine who has been trained under me for last approximately 10 years are performing the RT-PCR as well as the what do you call is the antigen tests for COVID-19 in this our region. So we are having a tremendous pressure for performing within limited resources. Now, when I speak about limited resources, it has in this thing, the as a biologist or as a bioinformatician, we should always have a thought that how to improvise or how to do innovative and valuable research along at the same time when you are having lack of funding. So I must tell you in such a way that you should not be having lose your moral, because everybody, most of the people will suffer from lack of funding. The usual fellowships, scholarships which were being awarded to people from developing and underdeveloped countries as well will have a rapid cut. But at the same time, we must not lose our hope, and we should skill ourselves in such a way that within our limited resources, we should have enough courage to do valuable research. At the same time, the sponsor who are sponsoring this HackBio internship, I must congratulate them for their timely intervention for the sake of science and for the sake of biology. And they have collaborated with so many people from so many diverse backgrounds. And at the same time, there is no financial burden for the student. That is a great thing. At the same time, I once spoken to the people also. I had a kind of commitment towards incorporating a little whatever to this kind of causes, and I'm always open to this, and I'm very much fortunate that I'm having this opportunity to interact with you all. At the same time, I shall also tell my interns for the last the 5 years back, the situation was entirely different when people had plenty of opportunities, when the governments as well as the international bodies, UNICEF, and all big universities were providing a lot of funding for research. We also had a very good time. At that point of time, we had opportunity to interact with the people in very good institutes in person. It was not all virtual meetings like these days we are having. So those opportunities may be lost, but at the same time, I should tell all the interns as well as whoever are there that in these situations also, we should keep on our work. And it is not that if we do not have access to big laboratories or big equipments or big softwares, we must not lose hope, because in there are some good tools for wet lab experiments as well, and for the research in bioinformatics as well, which you can use in a limited cost and limited investment also. So I think that the all the students and all the interns should explore a little more if they want to do on-site training, on-site opportunities if they are looking for. They may have a little difficulty, but not for everyone. Not for everyone who those are having a particular skill set which the industry needs. They will definitely be placed. In my country also, this is the same phenomenon where we feel a lot of difficulty. We face a lot of difficulty to place our students to the exact domain where the student anticipate to get in. So at the same time, this is a situation where you will have plenty of opportunity to to play with your skill sets. You may acquire entirely different kind of skill sets because I have seen in the group also there are some botanists who have worked all their lives with green plants. They are also encouraged to get into bioinformatics. Why? Because generally bioinformatics have an opportunity to result into good research instead of very limited funding. So it's a very encouraging thing that interns from various disciplines have participated, and I'll request them to learn from whatever from their mentors. They guide them and do the hands-on and use the internet in such a way that they can result into good research. One more point I want to elaborate is that there are some opportunities in the industry which are rebranding themselves at the same time reshaping themselves. For example, there will be opportunity for a bioinformatician in environment. There would be an opportunity for bioinformatician in waste management. Then it is not that the bioinformatician will always look for a vaccine, will always look for a drug. They have plenty of other responsibilities as well. Okay, it is quite understandable that the clinical research and biological, that means the applications of bioinformatics in clinical domain is very prominent. But at the same time, we can also explore other domains. For example, as I have said, environment, then waste management, and then increasing yield of food grains. Although I am a clinical person, but I can understand that increasing the yield of food grains and to produce more valuable food in those domains also bioinformatics can be used, because as everybody of us knows that there will be thus in the screening phase bioinformatics will help you to do a lot of background work with very minimal cost. For example, if we can design some salt resistant enzymes. For example, some enzymes are there which is needed for the synthesis of plant hormones. Now, those plant hormones, if those are not there in the plant will not grow. Now, the problem with the hormone is not that the hormone is a molecule for plants which is not a protein, but the enzymes which produce those hormones are proteins. Now, these proteins have a threshold up to in terms of salinity, in terms of many other things. For example, pH, in terms of temperature and many other things. So if we could design or if we could engineer those enzymes which are responsible for the plant growth to sustain certain kind of stress by using bioinformatics tools, not very hi-fi tools, very simple tools. Only that you have to put a lot of effort, you have to put a lot of time in those using those rudimentary, if you say, tools. But I'm sure that if a person is diligent enough, he can definitely able to find out what do we call as a solution for a given specific kind of problem. The same time, one more important thing is interns should focus on studies uh, which are relevant to the society of the person who around the locality. For example, for myself, I will not do. I would not like to do something which is very much irrelevant. It may be of much interest to me, but if it is highly unrelevant, or if it is do not have any impact to the people I work for, then I think we should, for some time being, we should refrain from this kind of research. So then what is a valuable research? A valuable research may be small, but its result may be such that which will impact the fundamental requirement of human being. I myself belong to a developing country. It's not a developed country till now, and therefore I am also concerned about my people. The small way, if I could contribute. And likewise, for some other interns also, intern may have a excellent interest to the working of the brain. For example, if a person of physiology is very much interested to find the answer, why do we dream? Okay, or why do we speak lie? Or why do we meditate? Kind of this kind of concepts is very good for the whole humankind. That is for sure. But today we must focus our studies, or we must focus our resources in those ways which will, at least for time being, will help the human being to sustain the challenges it have in immediate future. I'm not telling the people to lose their imagination at all. I am not advocating that you should be less innovative. It is not that. It is only that because you know, since everybody of us are having some limitations of resources and many other things, we we should channelize our energy in such a way that we are not investing so much, but at the end of the day we get some good results which have some impact to the immediate need of the society, or the community you belong to. For example, a vaccine for COVID-19 is being tried for many days, for a lot of days. So if somebody anticipate that after 1 month of internship, you can design a fundamentally new concept, or you can design a fundamentally and excellent workable vaccine, sometimes it would be not possible. But what will be the possibility? The possibility will be if you can produce a database where you can identify why exactly some people of a particular region are less prone to the severe symptoms, the same time why in some region people are more prone to severe symptoms. So this kind of work will not need to have you a huge laboratory. This kind of work will not need to have you a lot of highly advanced software, hardware, things like that. What it needs a very meticulous, detailed study of existing literature, very meticulous and repeatable studies and uses of already established, very simple, rudimentary types of softwares. If we use those in a judicial way, in a planned way, and if we have our visions clear, I'm sure that we can definitely able to do significant work within all our limitations. The same time, I'd also like our interns to explore more. Previously, they would have searched for what shall we say is research funding for their research from their government. Now they should explore from other governments. They should explore for industries. They should seek funding from industries by saying that okay, we can have some value addition in your product. Yes, we can produce some sort of information, valuable information for your company, and if you provide me this much of funding, I can definitely able to help you. Likewise, also the interns should have to uh, do their or keep on doing their work. For example, for last couple of years in many countries, as I know, have reduced funding for basic research. But that is an opportunity, at the same time is a very big challenge. Very big challenge, because Nowadays it will be difficult for one to purchase or procure equipments which is needed, which he or she anticipates that it should have in your lab, but you will not able to meet those requirement of yours. So what you should do, you should stop working. No, you should not stop working. What you should do, you should explore for cheaper versions. You should explore how to improvise. For example, for in I have seen in hospitals in India, if you for a simple, very simple thing, for example, to find out that if a person is suffering from some kind of anemia, so the tests are such that it may cost approximately $1. But if you prepare the reagents at your lab, where the raw materials are only available, only you need to have some precision measuring pipettes and measuring cylinders. Generally these things are not available in a hospital, but in a nearby lab only it is very much available. So if you use your uh, if you use your ideas in such a way to instantly produce solutions with your small resources, then I think it is possible to do bigger work within the limited resources we have. So I also came to know from somebody that one of our intern has been infected with the virus. I expect a speedy recovery of her, and she'll be definitely a fighter, and she will come out with all her victory. If you have anything to ask, you can ask.
Okay, thank you very much, Doctor.
You want me to specifically talk on something?
No. Yeah, you've talked on the interesting things we wanted to hear, especially what scientists can do and the trying times we are in, and what scientists can do. We should not lose our motivation.
Mm-hmm.
We should not lose the scientific curiosity. So
Mm-hmm.
usually, this is the pattern we take. After introducing you, you talk, and then the students can ask questions. So we have
Yeah,
like
sure.
30 minutes. We have like 30
Yes,
minutes,
yes.
so they can. So usually, it takes a few minutes before the first set of questions begin to come in. So we are waiting for your questions. By the way, I wanted to ask a question myself.
Okay.
Okay. So can you tell us how, like, any, I don't know if you have any of the design of of the vaccines you have probably designed in the past, or how you went about the project itself from scratch
Okay.
to final,
Okay.
yeah.
Okay.
Mhm.
As the theory goes, and as the protocol generally people follow for design of a vaccine, is not a monumentous task. It will take a single person with some resources, 5 days maximum, to standardize a particular epitope. Now, there are problems. The problem is, if the epitope is so stable that it will repeatedly give you similar kind of results, then it is absolutely fine. Now, this thing happens with the viruses, which are DNA viruses, as I have said in the first day itself. Now, DNA viruses have very stable genome. They do not change rapidly. In fact, they in general, they do not change at all. They are more stable than a bacteria sometimes. For example, polio. Now, polio's epitopes are so stable that it is more 100 times more stable than the epitopes which are being expressed by Mycobacterium tuberculosis or the malaria parasite. So what happens is, in 1970s, the Savin vaccine was first designed. They had no software, they had no information of bioinformatics, they only had wet lab skills. Now, using the wet lab skills, what they did, they identified the virus first, and then they used a number of chemicals in order to induce a state of a state of what shall we say is lower the potency of causing infection, uh, and more, and to their own astonish, it was a formaldehyde, a very simple chemical which used to make the virus so so unharmful that it could be injected to a person by a simple needle. Need not to had any specialized drug delivery kind of mechanism or things like that. So that was the one story where once when the vaccine design was very simple. Now after that, when the bioinformatics part came, then many of the top bioinformatics lab produced pipelines. Now, what was the pipeline? In the pipeline, they used to identify a particular epitope, and then they used to identify the magnitude of antigenicity. That means how it will start interacting with the cellular and non-cellular part of immune system of human beings. And in the third part, they used to interact those epitopes with the receptor molecules, or more precisely, you should say MHC molecules. MHC molecules themselves are proteins. And most of the time, the vaccine or epitopes are also proteins or glycoproteins. Now, for protein-protein interaction, there are plenty of resources which will give you the binding domain in with respect to the particular amino acids which are interacting with each other for the ligand and the receptor. So this task became very, very easy. Now, once something is very easy, there is a dilution of the resource. Then what happens? Then plenty of useless papers came, which never resulted into a patent, but they produce a lot of manuscripts, lot of data with respect to the use of bioinformatics tools for vaccine design. Now, there are two aspects of it. If there are a lot of published work, it will help the next generation to establish a database upon which he or she will further carry out the work from that point. At the same time, the negative point is that you start using these resources only for the sake of producing papers. Now, in RNA viruses, the problem is, now you see this virus is very new, therefore we call it novel coronavirus 2019. But HIV is very old. So for HIV, if you search in PubMed, you will get hundreds of papers which have described the antigenic domains or epitopes which can potentially give you vaccines. Now, till date, has anybody developed or has anybody come up with vaccines? Bill and Melinda has spent a lot of millions and billions of dollars for vaccines, for clinical trials, for plenty of studies related to HIV. But there has been no path-breaking result. Why? Because the virus is so notorious that it will not remain stable. In fact, in different population, it have different what do you call come outs. In some populations, the patient with a simple ART will sustain for so long. But in some population, instead of using ART and all other things also, the sustenance of the sustenance of the patients are very less. So there is an opportunity to study most of the vaccines in terms of the population, the difference of the exposure or difference of the intensity of the symptoms. So instead of finding a cure by means of a vaccine, If we channelize our study to that way, it will be valuable. But at the same time, we should also keep on trying for designing vaccine. There is no there is no doubt in that. But I also propose that why not we do some kind of engineering. That means what? Engineering means we identify an epitope, then we keep on trying the amino acid sequence of that, so that at one point of time we find out a combination. Nobody knows it can happen. A combination which is like a magic bullet, which will produce a multi-subunit response. So if the virus mutates, still the vaccine will be working. But for that, we need to have a lot of work, and it will need to have a lot more work to be done in synchrony with many people in together and doing a pilot project kind of thing for that. So that could be done in future. But currently, if we are focusing for vaccine design, that is absolutely fine. But my idea was to identify the population, why different populations are differently susceptible to the particular virus. So that was my idea of my topic, that proteomics and vaccine design. It will definitely discuss on those things, why different populations are differently behaving with respect to the same virus. So we will try to explore that. And as far as my previous work are concerned, I have mostly not. I have always tried to use bioinformatics beyond the scope of docking. In many a times, it has been seen, and there is nothing wrong in that. The bioinformatics sometimes binds itself only to the experiments where some kind of docking is explored. But bioinformatics have for example, there are multiple softwares which have the ability to explore more the ligand receptor binding only, to produce much more valuable information than only to identify a pharmacophore. For example, if we do use a very good resource and use some typical target of COVID-19, and with the library of a huge database of molecules, we will at least find 10-15 leads. But do they ultimately lead to a medicine? Many a times the answer is no. So we have to find out why this is not happening. For there are some simulations where using an ADMET, you find out why exactly the drug is not behaving the way it should have. There are simulations where you understand how the drug is metabolizing inside the liver and then coming out to the system. So those things are already there. We are very much sure of that. But at the same time, it is very painful to see why many times the research do not end up into some kind of valuable patent. So if you do not have the resource for doing that, why not do a valuable research which will lead to some kind of new information, which will help the next generation to do the needful. At the same time, I'm definitely be using these excellent softwares which are been given to the students and to everybody by our excellent sponsors of this workshop, and that will be great to work with all those, and I'm very much open to that, and I have explored those softwares as well for my other work, where I was already involved in antifungal as well as antibacterial kind of studies, and we tried to identify solutions from very simple things. One example I can give you, in Indian subcontinent, the um, the diseases related to waterborne parasites or waterborne bacteria are very much prevalent. Now we are trying to find out very low cost solution for those kind of things. which will ultimately lead to some kind of patent in future. But at the same time, we are exploring bioinformatics to its maximum limit, to find out the interaction between those molecules and the whole gut biota of Indian population. So that is a different work that we are interested in. So I'll definitely look into all those things, and would like to collaborate with more people to find out what exactly is going on, why people are not finding any vaccine for this. If at all there is a possibility in near or far future also, if we could design a vaccine for COVID-19, for HIV, for all these kinds of RNA viruses, that will be great achievement for humankind.
Okay, thank you
Wale?
very much. Yeah, yeah. Thank you very much your for your response. I think you really dealt with it a lot. Thank you a lot once again. Um, why
I
is
wanted really some response from from the these things, our
yeah, yeah,
interns.
yeah. I
These
was
are all full
expecting
of my class only. Some other people are not there. I thought that there will be
yeah,
some other people as well.
it's supposed to be for everybody. I don't know. Some
Mhm.
persons left, maybe due to internet connection. I don't know.
Okay, anybody have to ask something? For example, like, oh yeah, why we are not jumping into the vaccine design pipeline?
Yeah,
So we
I
are not
think
jumping
most okay.
because
Okay, yeah,
in every
please go
step,
ahead.
yeah, in every step we are trying to add some kind of value. It could be a repetitive work, but this is the way I generally look into a problem. I do not jump. I previously do a little background, and then slowly, I wanted to reach there. And I am confident that our team, I think I am confident that in all other teams, they have done some study where they are good enough to publish some work. I think that these interns also, they'll take this opportunity to sincerely do some good research and try to publish in good journals, which will give a good impact to this whole work the collaborators are doing. Mainly Wale and Samson, who are coordinating the whole thing with so many people. Wale, I would like to ask you one thing. The total, how many mentors who are working on different projects?
We have a total of six mentors. I think
Okay,
five
okay.
mentors rather.
Okay, great. Everybody will be definitely doing. But do you, Wale, is there a thing where the mentors themselves can interact with each other? Is there a possibility?
Yeah, we created a mentors hangout channel, but
I see.
that's actually on Slack.
Okay,
I don't know. Yeah, we can. There's every possibility, probably.
okay.
But right now, I can't really say. Maybe I can't assure that it will be this week
No,
because
no, that is absolutely fine. Okay, and I would definitely like to congratulate the sponsors of the event,
yeah. Thank you very
who
much.
have no really, because it's a good idea that in the resource limited setup, how you collaborate and how you learn anything new in order to do some kind of valuable work. It is a excellent. I must congratulate everyone
Thank you very
that it's
much.
a good opportunity after the day. To 1 or 2 hour if I spend with some people, it only gives me good vibes, and it only enhance everybody is this thing. I feel it like this way. I may be old teacher, but I do not feel that I am not a student. I always keep on trying to learn new things. That is a good opportunity for everyone, and I
Yeah.
hope in coming years also you come out with flying colors, and you do internship for many students like this.
We will be speaking with one of the main sponsors by Friday. So she will be on a call, on a Zoom call. She has already promised to join us. So probably
Fine.
we can all say our big thank yous to her. Honestly. Hello, hello, hello.
Hello.
Okay, okay. Uh,
Yeah, I'm there.
I don't know. Nobody's asking questions.
I
It's
think it
strange.
is okay.
I think they are tired from the loads of work
Yes,
we are
yes, that
giving
could
them.
be. Mm-hmm.
Because it's I think it's towards night time in Nigeria
Mm-hmm.
and in most places all over the world. So maybe it's really difficult for them.
Mm-hmm.
But thank you very much for joining this call. We sincerely
Okay.
appreciate your time actually, because we know it's not. I'm also aware that you are supposed to have a meeting with your interns today, and you had to
Mm-hmm.
postpone it. We're very sorry for that. In
Mm-hmm.
fact,
No, no, no problem. Tomorrow we'll do it. I
we
have
have a
spoken
very
to some. I have said that they should keep on the way we have already planned,
okay.
and tomorrow we can do it. There is no
Okay.
absolute problem.
Okay. Someone said thank you very much, Dr. Surajit.
Yeah.
I'll let you written says thank you. And I think most of your students are also here. Okay. Eva said thank you, Doctor. It's been insightful. Okay, there is no question. We call it a day for today's Fireside Chat series. Thank you, everyone, for attending today's call. And we sincerely once again thank Dr. Surajit
Yeah.
for his time and for sharing significant insights with us. We really appreciate you. To everybody, have a nice day.